Yep. Okay. For the kind introduction. And thank you to you, Andrea, and all the organizers for bringing us all together for these 3 days. And also I congratulate you for the choice of the distinguished speaker. I mean, like you said, I moved from math to neuroscience working with Carl. That's why I think he's definitely the right person to inspire people from math to switch. So today I'll be talking in this short presentation about some recent work, ongoing work we're doing with colleagues who study depression, the group of Helen Mayberg in Mount Sinai. And they have one of the, let's say, success stories in neuroscience. They have treatment with DBS, with deep brain stimulation for depression. Treatment means that people with severe depression that are at the risk of committing suicide, they go, they have a treatment in the operating room, they have current thrown in a certain subcortical area called SCC, subgenual cingulate cortex. And then the depression symptoms go away. But this treatment does not work for all patients. Or there are some that works for a certain period of time, and then they relapse. So the question colleagues working and Helen have is why DBS works in some patients for some time and not everybody, or why there is relapse, etc. So I think we can bring ideas from math that, like I said, this is ongoing work, and I welcome feedback at the end and throughout the next few days on that. We can bring ideas like the ones we heard about and will be hearing from brain modes to try and say something useful in this direction. And yeah, to make the long story short, I think that one way to judge whether DBS is effective or to try and look for efficient treatment with deep brain stimulation is to look at the interaction between two levels of brain activity, not, let's say, go away from just looking at the spiking at the neuronal level and look also at the mesoscale oscillatory electric field level and look at how the two levels interact. And I'll come to that. And my claim is that by developing a theory for that, along the lines of what people will be talking these days here, we can get some new criteria that can guide us to efficient treatment. But I know this is a bold claim, so I'll try to unpack it in the next 15 minutes or so, or less, actually. So what do we know? We know that brain power measured before and after acute treatment. So you put a depressive patient in the operating room and you throw current to their brain and you measure the change in brain power before and after this acute treatment. So this correlates with the improvement in behavior, with how much behavioral, let's say, symptoms, depression symptoms go away. And this is what you see up here, the change in Hamilton score and the percentage drop in beta. This is work by Helen's group 2021. So an acute drop in beta power before and after, just before and after the first week, the operating room. And there's also this other work last year that you see over here, sorry, to learn, in Nature by Shankar, Alagapan et al., who showed the following. They used machine learning and they picked a combination of brain modes, meaning parts of the frequency spectrum in beta, in gamma, in different parts of the brain. They used the machine learning algorithm to say which ones have the most predictive power. And then they found that this particular combination found with machine learning was able to track, you see weeks on the horizontal axis at the bottom plot here, and you see two curves, the blue and the red. And the red is the machine learning spectral discriminant component, this predictive feature, combination of brain power features. And the blue curve is the Hamilton score, the behavioral score. And you see that it follows. So another, and crucial in this plot, the red crosses the threshold from, let's say, control or health to sick state 1 month earlier than the Hamilton score. So this means that when you trained the algorithm on patient data, you were able to identify using this predictive feature relapse of this particular participant that happens, let's say, in week 2017 or something, using the blue curve, the Hamilton score, you found that 1 month earlier with your machine learning feature. So it can predict relapse, essentially, when you use the machine learning to combine spectral features. So the bottom line here is that somehow brain power relates heavily with Hamilton scores and behavioral improvement, and it can even track them, as we see, for example, also in this plot over here. So this is, again, different subjects. This is a relapsed subject that I talked about before. It goes from healthy to sick state when it crosses this dashed line. The others, you see, they converge over a period of 25 weeks to below the line. So they don't converge, they are better. This is the data, by the way, we're using also for what I'll talk about. But I'm using basically the first month, so this part of the spectrum. And you see that there is some kind of fluctuation, right? The blue and red, they go somehow together, they go up and down across all subjects. So behavior and brain power also fluctuate after the acute treatment in the operating room. And this is what our, let's say, the phenomenology is. So these brain power fluctuations that we measure with our invasive electrode, they come from a particular area primarily, SEC, this subgenual cingulate cortex, which is basically the target for DBS, where we apply our electrode. But also it's important for depression because it's associated with symptoms, basically, like mood dysregulation, excessive rumination. You see here different images, recordings of glucose consumption, blood flow increases. So there's usually a hyperactivity in depressive patients. And on the bottom here, by the way, you see when you apply DBS in the operating room in this particular area, what the protocol is. You have 2 windows during one day of 30 and 20 minutes of stimulation on and then stimulation off. And then what we're analyzing is the month that follows this stimulation. So DBS is known to normalize behavior also in animal data that I show here. You see mobility ratings and anhedonia rankings here from animal studies, and you go back to healthy, let's say, state just after this acute treatment. So the question now is how to describe these brain power fluctuations that somehow relate to behavioral improvement, and then what drives them. So we try to understand basically how to somehow control these fluctuations, or try to distinguish whether they are pathological or healthy. So we need a theory for that, and we'll use some of the ideas we heard already in a very nice presentation by Axel, Lyapunov exponents and slaving principle and all this. So I don't need in this building talk a lot about Lyapunov exponents. This is our way, or one of the ways we use to characterize, of course, brain activity under the assumption that we're close to some stable stability threshold, some stable attractor state. And this is a common assumption we do when we use dynamical systems approaches to cognitive neuroscience, decision making, or memory research, etc. So the idea here is that when we apply DBS somehow, this neuromodulatory input, we will be moving from, let's say, some stable attractor state in the phase space diagram along a stable or unstable direction for different patients, right? And then we will characterize this itinerancy, this movement between attractor states using Lyapunov exponents to begin with. So that's the approach. But then crucially, like I said earlier, the trick here, the twist, is that we're not going to be looking only at Lyapunov exponents that characterize spiking activity, neural activity, but also we will have an observation model on top for the electric field from basic electromagnetism. I picked a model here based on the available data we had. So we'll have 2 levels of description. We'll have a neural field model level, and we have a lot of neural field people today. It's great. And I think for the next few days. So this is why we're using neural field now to predict spiking activity, plus a model of the electric field on top. And the idea is that DBS will perturb both these levels. And also information presentation more broadly lives at these two distinct levels of electrophysiological responses. And then the efficient DBS will be the one where the field basically, because DBS affects the electric state, right? The energy of the neural circuit. So this means that the electric field and the neural activity will be going back from, let's say, pathological attractor. That's the hope to some control-like attractor. So this is all motivated by a new kind of principle for brain function we introduced with a couple of colleagues, with actually Ed Miller and Gene Friedman a couple of years ago, called cytoelectric coupling. So this is just to motivate where I'm looking at these two levels. I think it has some theory value. So the idea here is I want to find efficient DBS, right? And I claim efficient DBS is when the energy state is efficient. And this assumes that what drives this itinerant dynamics is the electric field. So DBS, what it perhaps doing is it perturbing to a new state post-OR, and then guides neural activity in the same way that a conductor, you replace the conductor in an orchestra or the football coach in a football team, and then the team, the same players, the same orchestra will play the same song differently. That's what we're trying, let's say, to do here. And of course, the electric field, of course, will be affected by the altered neural activity. So there is this circular causality we heard already. So this principle is just an extension of Ephaptic coupling that you might be familiar with, and says that the conductor of the brain might be the electric field that helps organizes neurons and crucially the whole cytoskeleton structure. So the thing that binds neurons together and filaments and proteins and enslaves all the spiking activity in a top-down fashion. And the goal is, of course, to have efficient information process. So we can now essentially write down math equations, and also as many, but I've skipped lots of material here, but happy to discuss, and they're in the papers. So basically, you can have a neural field model, you can have an electric field model, you can expand into some Fourier eigenmodes that we now like in this room and get equations for circular causality. So the psi, one dot here, is a temporal evolution of the extracellular potential, and this depends on the boundary values of the potential on the cortical patch that is affected by DBS. So DBS changes these boundary values denoted by superscript 0 here, but then, of course, the enslaved spiking mode, the psi for transmembrane potential, is also a function of these boundary values. So DBS, by altering this, alters both psi, the source, and the temporal evolution of the extracellular potential. So we have this circular causality. So we now calculate, as promised, the Lyapunov exponents for these two modes of activity. And this is what you see on this plot. We have 4 subjects. Remember, I was focusing not the relapsed. And then we have Lyapunov exponents on the vertical axis, recordings here shown for the first week after OR. We have red and blue curves for neural activity and electric fields. They point up or down, positive and negative. This plot is the same as this one, where I have split the Lyapunov exponents for neural activity and electric fields, and they're light gray and dark gray, depending if they're positive or negative, just to show the transitions between stable and unstable states during the period after OR. So this is basically the information here is the same in these two plots, and the same for others. And then the yellow and red arrows, what they show is transition from stable to unstable, let's say, directions in the phase space. And we have, of course, some similar number, but crucially, of yellow and red arrows, crucially, the blue bars, as predicted by our model and the slaving principle, are larger than the orange bars. And this speaks to the cytoelectric coupling I was talking about, that neural activity is more unstable than fields because the Lyapunov exponents are larger. And you can also see that when you write down the math equations, besides calculating from data, right? But I showed this data-driven, this is the model. And here, this is basically the quasi-static approximation that is common in brain electromagnetism. So what is the bottom line here is that neural activity is more unstable. So the field has this stabilizing conductor-like effect. That's one thing. What else? I claim that somehow this brain power, the fluctuations we observed earlier, that we know are in our data, somehow show itinerancy. So the question now is, does the brain power correlate with basically Lyapunov exponents? So that's what we found. We found that indeed, itinerancy described with Lyapunov exponents correlates for each subject across the month after the operating room with brain power. So indeed, itinerant dynamics are linked with brain power fluctuations. That was a claim I made in the beginning, right? But somehow there is some meaning there. And then somehow this itinerancy relates also to behavioral improvement and Hamilton scores. That's one thing. The other thing that I jumped a lot of things here, and I would need a longer talk for that, is to show the top-down guiding effect of the electric field. So what I did in a nutshell, maybe you get an idea, is we have a neural field model, right? And then that predicts the same LFP data as the recordings. And then we have an electric field model on top. And then we use Granger causality to look at the influences between the spiking activity in our model, in the neural model, and this electric field activity at this higher, let's say, mesoscale level of information representation. And we find indeed that the influences, the top-down from the field level to the neural activity level, the blue, are much larger across all subjects compared to the reverse direction from spikes to electric fields. That's one thing. So you can say that the Ephaptic coupling drives this itinerant dynamics. And then the other thing is that the strength correlates also with brain power. So there is a link between the fluctuations we see and these top-down electric field effects that we somehow inferred from our data here. So this answers, in a sense, the second question, right? We described our itinerant dynamics, and we say that one guiding force is the aggregate electric field. And this is basically our conclusion here, that information seems to propagate from mesoscale to neural activity to the guiding itinerant dynamics, like I said, the Ephaptic coupling. And why this is useful, right? That's the last part of the talk, because people in neuroscience, they're not interested only in math. Maybe many of us are in this room. They are interested also in people like Helen, in how can I help my patient? How can I do that? I'm not saying I have a treatment. I'm saying I have an idea how to find better biomarkers for depression. And Ephaptic coupling strength could be one. We have a couple more. I'll talk very, very quickly about this. But the bigger picture is what we try to say here, that if electric fields guide neural activity, this could be particularly relevant for treating depression, because depression and DBS treatment will be successful when it manages to alter the brain's electric field and move it away from an electric field and consciousness state, because a field for many people defines a consciousness state, the pathological or depressive state versus control, that underlies depression symptoms towards a state similar to that found in controls. And this is not, just some abstract, let's say, hypothesis. We have some way of making it concrete by introducing measures of combinations of Lyapunov exponents at these two levels. I don't have time now to go into this detail, but I'll show you just one last slide with results. What are these? 8 panels, 2 for each subject. This is subject 905, left recordings, right hemisphere recordings, same for all subjects. Each panel has 2 lines, the red and the green. The red line is our first Lyapunov-based measure, which is instability frequency, essentially how often we have unstable directions in the phase space for both neural activity and electric field. And then the green line is this other measure, which is the wandering time difference. Again, how frequently they move around and they change basically from stable to unstable states. But this is the relative difference, like when you have the conductor and how well they, in a sense, synchronize with its musician. Now, crucially, what happened here is that we were able to parse the Hamilton score space, at least these are first results, right? Using these measures into people having low depression versus high. So on the left, these two subjects, 905 and now 910, have low Hamilton scores. These have high. And if you look for the low patients, this red measure is below 20%, while for the others, it's above 20%. This green measure is negative versus positive in people low and high, but not everywhere. In left hemisphere only. And I showed that to Helen. I said, okay, I found this. It makes sense if I look at the left hemisphere. Is left hemisphere recordings important for depression? He said, yes. He came up with a whole list of different results. Well, when people look for treatment of depression, they look at activity left. So, for example, you have an asymmetry in PET studies. You have neuromodulation target, this SEC in left hemisphere. You have this spectral discriminant component, spectral feature is also constructed from measurements in the left hemisphere. The left simulation evokes higher, more consistent whole brain responses. You have physiological measures in heart rate. Again, left. So there is maybe some value to what we have found, and this is ongoing work. Got some funding. Let's see how it goes with all the things changing the world. And thank you. And Helen, oh, sorry, I did something here for your attention. And